Hello, welcome to Pen with Love. I am Tutor Additional.
Some people have been having difficulties in attempting practicals in biology, and this is the reason because of the ongoing Waec examination, which very soon they will be writing the biology practical. We have decided to look into some specimen analysis according to the 2026 Waec practical biology examination. Mind you, this video is not in any way for examination malpractices. Examination malpractices is a crime everywhere. Therefore, we urge all our listeners, students, and everyone to stay away from any form of examination malpractices. Now, we are going to make this very short, but do well to try as much as you can to stay tuned from the beginning of this lesson to the end, because it will really help you in your practical biology. Please note, every analysis and questions raised or treated here are strictly based on Waec past questions in related to this year Waec specimen. And I want to guarantee you, if you practice all these things very well, watch from the beginning to the end, and you pay attention, you are going to have a very easy examination when it comes to biology practical. Looking at the specimen given to us, we have specimen A to have, excluding some alphabet like I. Now, let's look at those specimens one after the other. These are the specimens you are seeing on your screen, and we are going to be showing the pictures related to each specimen as we dive into the analysis. We have specimen A to be groundnut pod containing seed, specimen B containing crushed roasted groundnut seed, specimen C beaker with 20 ml of water, specimen D beaker with 20 ml of table salt solution, specimen E beaker filled with sandy soil, specimen F portion of alimentary canal of a small mammal showing esophagus to the end of the stomach, specimen G branch of a plant with mistotube, or let's say a plant with a parasite, specimen H evaporated milk, specimen J fresh fish, specimen K, mind you there's no specimen I, specimen K dry maize grain, specimen L tomato paste in petri dish, specimen M fresh tomato fruit, specimen N rotten tomato fruit, specimen P thermometer, specimen Q meter rule, specimen R quadrant. Then these are the specimens given. We also have some reagents given, such as sodium hydroxide solution, such as Benedict solution. Another thing that we have, and we are going to be make mention as we are moving forward in this video. Now, for this practical, I will streamline it into 5 to 6 aspects in which I want you to pay attention to. According to those specimens given to us, there will certainly be food test, because from the groundnut, from the milk, from the tomato, and from the maize, we can have tests for protein, we can have tests for reducing sugar, we can have tests for carbohydrate, starch, we can also have tests for fat and oil, according to what we were given and those reagents I have mentioned earlier. Also, we can look into ecology, and I will have put this together, but I just separated it: ecology and measuration. Now, under ecology, we have the quadrant, we have the mysto2, the parasitism, that is the parasite plants. Then we also have the sanding soil, which when you look at those things, there are questions that is going to come out under those likely to be in ecology. Another thing is the measuration. The thermometer, the meter rule, they are used for measuration. We are going to talk about that too. And number 4, we suggest we look into mammalian anatomy, the alimentary canal given to us. Then we have separating techniques, the separating techniques, because we have water given to us, we have sanding soil, we have saltwater solution. There may be a kind of mixture which we are going to be needing to separate. And last but not least is the aspect of drawing, the biology drawing, because there's no way you are going to have practical biology without drawing. And according to what we are given, you are even given a hand lens as part of the other materials supplied to you. Now let's take them one after the other. Let's quickly look at those specimens that they are on their own, that we can look at some questions that can come under those ones. Like specimen A now, granite pod. Specimen A, granite pod, and this is the diagram being attached to the slide here. So let's look at the possible question that can be asked under this. Mind you, in biology, don't get yourself scared. Don't give yourself any headache. All specimens are there for you to observe. If you find anything strange or unfamiliar, just take your time to observe it, look at it critically, and you'll be able to answer any question given to you, I assure you. Now, the first question that can be asked under specimen A is identification. All specimens given to you will be asked to identify. So, when you are asked to identify according to the way Waec used to put their questions, so they can ask you to identify specimen A to P, specimen A to R or Q, whatever. So, when you want to identify specimen A, the answer is on the other side here. So we say specimen A is granite pod with seeds. So mind you, don't just write granite pod, because the one you are asked to provide is not empty. So make sure you signify there that it's with seed. Identify the fruit type. What type of fruit is specimen A, granite pod? Let's say you are asked. So specimen A is a leguminous plant. It is a legume. So another question they can ask you is for you to state observable features of specimen A. When you are asked to state observable features, mind you, from the word observable, observe, those things you can see with your naked eyes, not those things you just think should be there. So when you are given your answer, make sure you give answers based on what you can see with what you were given. So according to the pictures we have here now, observable features we can bring out from this is it has a brown color and it is dry, it is a podded fruit, it has rough outer surface. You can see all these 4 points, they are something that can be observed. You can see them, know any other thing, and any other thing you can observe from that specimen given to you, you can add them to it. Now, another question they can ask you is state the economic importance. Economic importances of a particular specimen can be asked. When you hear the word economic importance means everything that the specimen can contribute to the society, to the ecology, to the ecosystem rather, to the environment, to anything. So maybe the good, the bad, anything, either positively or negatively. So these are the aspects. Now let's look at the economic importance of specimen A. Economic specimen of specimen A: source of edible oil, source of protein, used in livestock feed as a raw material, they can also use them as a source of income. So they can also ask you to classify specimen A. When you're asked to classify specimen A, majorly at this level, they normally ask about the kingdom, the phylum or division, and probably they can ask you the scientific name. So these are the things you have on your screen. The kingdom is plant A. The kingdom is plant A. And the division, or that is popularly known as phylum, but when it comes to plants, you call it division. The division is magloliphyte. So the genus is Arachis, and that of the species is called Hypogea. So in a nutshell, we can say specimen A, which is groundnut pod. We can say the scientific name of groundnut is Arachis Hypogea. So when you are writing scientific name or botanical name, when it's for plants, you can call it botanical name. Make sure, when you look at the one you have on your screen, Arachis Hypogea, you can see it is written in italic form. The first letter, the first word, which is the generic word, is written starting with capital letter, while the second one is written without capital letter. That is how it must be, and there must be gap between the two. There must be space. You cannot write in italic form. Therefore, you will need to underline. So you underline Arachis and you also underline Hypogea differently, not together. Not together. So they can also ask you what type of germination does this specimen undergo. They undergo hypogea germination. That is, their seed, when they are germinating, their seed is below the soil. So the shoots come out of the soil while the seed remain in the soil, unlike epigea and others, which their seed come out when they are germinating. So about specimen A, that is that about that. Now let's look at specimen F, the alimentary canal of a small mammal. The questions that can be asked under this are listed above in your screen. Identify specimen F, name part present in the specimen. They can also ask you the functions of the parts mentioned that you give answer to. They can also ask you what are the adaptation of the stomach of the specimen. Now let's look at the answers to those questions. If you are asked to identify alimentary canal of a mammal, you don't know the kind of mammal, you don't know the type of animal they have given you. So you are going to just say that alimentary canal of a mammal. That's all. Now, what are the parts present? Don't mind according to the specimen, they said from the esophagus to the, so when you are identifying the specimen too, you identify alimentary canal of a mammal into bracket esophagus till the end of the stomach. Mind you, that is where so when you say alimentary canal, it means the food. So make sure you signify and you signify the parts given to you. The major two parts we have there is the esophagus and the stomach. But we also have the cardiac sphincter, which is the muscles that always hold food tight after the food has gotten into the stomach, so that it will not be going back to the mouth. And we also have the pyloric sphincter. The sphincter is also that one that also holds the food tight, so that it's going to stay in the stomach for some hours, so that it's going to be fermented and also gets digested to some extent, mixed with hydrochloric acid and some other things inside the stomach. So they can also ask us the functions of each part, majorly the esophagus and stomach. Right? The esophagus is majorly a passage for food through the process, through a movement called peristaltic movement, because of the some ciliated tissue that we have on that in the passage itself. So they transport food by peristaltics. So the stomach stores the food temporarily. It also digests protein because of the presence of the gastric juice. That the presence of. It also helps to digest protein because of the production of the enzyme like uh, renin and pepsin that is present in the stomach, which helps to digest protein. It also helps in mixing food in the stomach, which turn into the child, then and it continue its further digestion. And another question that's going to be asked, that may be asked rather, is adaptation of the stomach. Look at the stomach. What are the things that make it to adapt? The first thing is the thick muscular wall. That is why it's going to be churning the food because of the muscular wall. The another thing there is the presence of gastric gland. The gastric gland produces gastric juice that secretes acidic medium for the digestion and the perfect work of the enzymes. There is also have expandable wall, so that when you are eating, it's going to be able to accommodate the food and water, and here you are taking. So let's move to the next one. Mesotu and host. A plant branch with a parasite plant. A plant host with a parasite plant, and that parasite is called mesotu. So that is specimen G. Let's look at the relationship. What is the relationship between the parasite and that of the branch of tree or the branch of plants attached to it? Looking at this diagram now, you will see the green part with full grasses, the leaves which are full. So that shows the mesotu. And when you look at the other part, you see a branch of tree with its own flower. The leaf of that flower branch is quite different from that of mesotu. So that is to show that we have different two plants here. So the plants that is on the other one is called mesotu, and this mesotu, the relationship between the mesotu and that of the branch, the plant itself, is called parasitism. Parasitism, I believe by now on this level, parasitism is the relationship between two organisms in which one is benefiting while the other one is being harmed. So the host is going to be harmed while the parasite is the one benefiting. So which organism benefits? The mesotu, which is the parasite. Which organism is harmed? The host plant. We don't know the name of the host plant. We are not given the name of the host plant. So you don't need to mention any name here. Another question they may ask you, type of parasite. Which type of parasite is mesotu? Mesotu, as you can see in the diagram below here, it has green coloration. The leaves has green color. That means it has the ability to manufacture its own food. The chlorophyll in its leaf can make it manufacture its own food. Yet it's still harming other plants. You understand? So it is partial parasite, that is hemiparasite. Hemiparasite. They can also ask you importance of mesotu. Source of food for some birds. Some birds feed on the fruits, the flowers and other things, and also helps for ecological interaction study. So one of the things that make it adapt and survive on other plants is because of the root type it possesses, which is Osterium. They can ask you that what is the type of root it possesses. It's called Osterium, or they can even ask you what make it survive on other plants, that is the host. So let's look at another specimen, specimen J. Specimen J is a fresh fish. A fresh fish. So a fresh fish, they can ask you to identify the specimen. Just write fresh fish. We are not given any name. You are not given, but if you can identify the name of the fish, maybe tilapia, maybe mackerel, just write it. A fresh fish, open bracket. If it is tilapia fish, if it is mackerel, just write it there. If it is catfish, it's fresh, just write it there. Now, observable features, like I've said earlier, when you're talking about observable features, you're talking about something that you can see with your eyes, something you'll be able to observe while looking at it. You don't need to be thinking of anything, whatever anybody have said about it. What you can see, like now they have streamlined body, the streamlined body to reduce water resistance or pay. So they have fin, they have the processed gills, you can write operculum too, presence of operculum covering gills. Then we have scale present on the body. If they give you a fish like catfish, that means your own will not have scale. You will not have scale present. But if you are given a kind of fish like tilapia and some other fish that have scale, you have it there. They have thin fin too. So and more of the thing, presence of the eyes is there, presence of nostril. So a lot of things are there to be observed on a fish. Like I told you, what you just need in biology practical is just careful observation. Careful observation, and you don't have any problem. Another question they may ask you is adaptation for aquatic life. They can even ask you the habitat of the specimen, and the habitat is aquatic habitat. So what are the adaptive features or adaptation for aquatic life? The streamlined shape reduce water resistance, gills for respiration. Don't just write gill, don't just write fin. No, gill for respiration, possession of fin for movement and balance, possession of scale for protection, possession of lateral line to detect water vibration, and so on and so forth. Possession of eyes for sight, possession of gill for respiration, possession of mouth for feeding, and some other things like that. Make sure you don't just write the part, make sure you write what they are used for when you are writing under adaptive features, because we want to know what they are using to survive. Now let's move to the question number 4 that is likely to be asked: economic importance of fish. Economic importance of fish. It can be source of protein. It is source of protein. It's not even can be. Fish is known as source of protein. Fish serve as income for people that are farmers. It also serve as export products. We use it for foreign exchange. It also serve as food for human and even some other animals. They can also be used as raw materials for some companies producing some finished goods. So another thing they can ask you on fish is classification. And when you look at it, like I told you, majorly the question normally asks, when you look at Waec very well, they used to ask question on the phylum, kingdom, phylum, and probably they can ask of the species and the generic name. But when you look at this now, the kingdom is Animalia, the phylum is Chordata, then the class, they can ask of the class is under Pisces. The generic name is Tilapia if you are given Tilapia. Then if you are given Tilapia, you can write the scientific name for it is going to be Tilapia spermani. That is, generic name will be Tilapia, while the species name is going to be spermani. And like I've explained here, how to write scientific name when we are talking about the groundnut the other time. Now the next one, let's look at specimen M and specimen N. They are almost of the same thing with just small differences. Specimen M is fresh tomato fruit, while specimen N is rotten tomato fruit. You can see both are tomato fruits. So we want to look at them. So these are the likely questions that might come out. Identify specimen M and N. Just like I've told you, they may ask you to identify all specimen. So specimen M is fresh tomato fruit, specimen N rotten tomato fruit. Now let's look at the observable differences. What you can see, observable differences. What you can see. So number 1, fresh tomato are firm, while rotten tomato are soft. Under fresh tomato, they are bright red, the color is bright, while under rotten tomato, the color is dull. The color is going to be dull. Fresh tomato will smell pleasant, while rotten tomato will smell offensive. Fresh tomato skin will be intact, while rotten tomato will be damaged skin. So looking at the pictures, you can see all what I've mentioned now are something we can see with our physical eyes. Now, another thing they can ask you is causes of rotting or causes of the condition of specimen N. And this is what we can answer as below. The first thing that can cause it is fungal attack. Fungal can be caused by fungi or bacterial attack or poor storage, and it can also be caused by mechanical injury, maybe during harvesting or when they are putting things together, there was a damage which causes it to be rotten. So they can ask you, how can we prevent this so that we are not going to have specimen N? You can refrigerate it, proper storage, dry environment, good handling, and some other things you can think of that we can use to prevent rotting. Another question they can ask you is which type of fruit is specimen M and N? They are drupe. They are drupe. They are drupe. And they can also ask you what are the economic importance of specimen either M. I think they can ask this on specimen M. So source of income, source of vitamin, these are raw materials for some people also use them in cooking and some other things like that. So they can also ask you to classify tomato. They belongs to kingdom Plantae, they belongs to division Angiosperma, and they belong to genome Solanum and species Lycopersicum. So in a nutshell, we can say the scientific name of tomato is Solanum Lycopersicum. Now, having said that, let's move to the next one, which is specimen R. On specimen R, the quadrant, the questions that are going to be asked on that is not going to be that much. It can be made of, it can be a wooden quadrant, it can be a metal made one, it can be a plastic made one. So quadrant can be made of those. It can be of a rectangular shape, square shape. It can be of a circular shape. So the one given to you is going to be the one you are going to mention when you are identifying your specimen. So I just wrote quadrant here. So the one given to you now be attached. Maybe a square quadrant, maybe a circular quadrant, maybe, you understand, a square wooden quadrant. You understand? So you put it like that. Then uses, what they use it to do, they use it to estimate population density. They also use it to estimate abundance. Then it's also used in studying plant distribution. Another question they can ask on this is, why is it suitable for plants? Why do we use it for plants? It is suitable for plants because plants are stationary. When you throw it, the plants will remain, and the plants will remain like that, and you can throw it more than multiple of times. So if it is animal, if you throw it now, the animal you find there, and you throw it to the same place again, you may not find such animal again because they are not stationary, just like plants. So these are the three likely questions I think it can be asked on that. Another thing is thermometer and meter rule, which I classify them under measuration. So under that, I have the picture of the thermometer here, but I don't have the meter is just the normal meter rule, the normal ruler we are familiar with, and when you see it, you sure identify it. So they can ask you to identify the specimen. So what are they used for? They use for instance, now thermometer is used for measuring temperature. It's used for monitoring environmental conditions. While meter rule is used for measuring length, width, height, measuration in a nutshell. So now let's move to the real work in this practical specimen, the main work there. And this is food test. Your food test is going to be in the following: specimen B, certainly; because you are given filter paper 2 as part of the materials that's going to be provided. So specimen B certainly, specimen K, and specimen L. Also specimen H. All your food tests cannot be more than all these 4 according to the materials given. And that is why you can see I wrote out those specimens that are likely to be checked, like the specimen A, granite pod, and specimen B. They are together. They are to test for one thing. Specimen H, that one is for protein test. Specimen K for carbohydrate or starch. Specimen LM is for that N is not even necessary. That N is not necessary. So LM be testing for they can also have K2, reducing sugar. So let's look at how questions can come under food test. Testing B for lipid. So you're going to have 3 table: test, observation, conclusion, or your inference, whatever. So but it's normally come with conclusion in biology, unlike in chemistry that we normally have inference. So under your test, they will ask you to play a sample of this specimen B on a filter paper. Then they can ask you to skills, so that then you observe. Observation is going to show that there is permanent translucent grease spot on filter paper. So that is what we call translucent test. And your conclusion will be oil or fat is present. When you look at it when you place the paper up, you will see the translucent side in the paper. So that's to show that oil is present or fat is present in specimen B. Specimen K can be asked to test for starch. So that is why your iodine and your white ties that is given to you as other materials, like I've said, that we are going to mention those materials as we are going. So when you crush specimen A, they can ask you to use specimen K. They can also ask you to use specimen L to test for dish. So when you crush it, you crush specimen K, they can ask you to add small water. We are going to we have one specimen that is water. So they can ask you to put it on, they will not ask you to put it on the white tie, then you add iodine solution. What is going to be your observation? Blue-black coloration. Blue-black coloration. And what's the conclusion? Starch is present. Starch is present. Another test is going to be on specimen L or specimen M. And they can also ask you to use specimen K again because of Benedict solution. Iodine is used to test for starch, while Benedict solution is used to test for reducing sugar, such as the fructose, all those monosaccharides. So they are there as a reducing sugar. Any reducing sugar can be tested using Benedict solution. Although we don't have any materials to heat, but they may also ask or put it there. So your observation is that when you add Benedict solution to any of the specimen, it's going to give you a brick red precipitation. So under conclusion, reducing sugar is present. Reducing sugar is present. So mind you, iodine is used for testing of starch, while Benedict solution is used for testing reducing sugar. The next one is your specimen H, which is known as evaporated milk. They can ask you to take specimen H, add sodium hydroxide, add copper sulfate. So when you add those things, observation, purple or violet color is going to appear. Then what is going to be your conclusion? Protein is present. Another thing that may be asked is under separating techniques. Specimen C, your water. Specimen D, your table salt solution that has already been mixed, already been mixed with water. And specimen E, sand. They can ask you to separate mixture of specimen C and E when they mix water with sand. So when they mix it together, the best thing you can use to separate it is filtration using filter paper, funnel and filter paper. Then after that, you can also use decantation in case they ask you more than one method that you can use. Another thing that can ask to be separated, or they can ask you for the method of separating them, is when they ask you to what is about specimen D, like water solution, this salt solution. You can use evaporator dryness to separate salt from water. When salt is mixed with water, you can use evaporation to dryness. So we are coming to the end of the discussion gradually. Let's look at other aspects in which they can ask a question. The other aspect is drawing. And the two specimens I believe the drawing can come from is specimen J and specimen M. That is the fish and that of tomato fruit. So for the fish, when they ask you to draw, make sure you draw very neatly. Don't shade, don't do anything artistic. It can be, if you ask you to draw, then look at the position you are asked to draw from. Maybe it's a dorsal view, maybe it's ventral view, maybe it's posterior view, anterior view, whatever. So then draw in a simple diagram. Then specimen M, the tomato fruit, you can be asked to draw longitudinal section, which you are seeing on your screen. And also they can ask you to draw in cross section, which is known as the transverse section. So you can try looking at this diagram to study what to draw. Mind you, no shading in biology drawing. Now, before we end this class, let me quickly chip in few tips that we can look into in biology drawing. In biology drawing, you are not to shade, no artistic drawing, but simple drawing. Drawing title, you must have your title below, then always label in one side of your drawing. Do not label scattered, labeled one side. Erase neatly. When you make mistake, try as much as you can to erase neatly. Label lines must not cross each other. Make sure your label line is separated and are parallel. Maintain uniform thin line. Don't draw with thick or shaded line or broken line. Then never use pen. Anytime you are in biology to draw, never use pen. Use pencil, HB pencil or 2B pencil throughout. Always draw to the given dimension. If you are asked to draw between 9 to 10 or 10 to 12, make sure you're drawing long as much as the lowest number and is not longer beyond the highest number. That is, if they give you 8 to 10, it must not long than 10 and it must not be smaller than 8. So don't forget your magnification too. Maybe multiply by 3, maybe 2 times of the specimen, maybe lower. So you have to indicate that. There's going to be a video specifically going to be on the biology drawing in the future. So another thing we need to know is that all the specimen, the food test, the measuration, the identification of specimen, the ecology aspect are only things that is going to be given within this specimen. They are not going to ask more than that. So, in a nutshell, we have come to the end of the lesson. Try as much as you can to go through them from A to Z. Write it out, practice them, study them, and like I've said at the beginning of this video, please desist from examination malpractices. So, I hope you have gained and you have learned something that's going to help you write your biology practical with ease. Finally, please and please, even when you see any specimen unfamiliar, just take your time to observe. Identify any features you can see in there. If you have any other questions related to this specimen or to any other topic in your biology, in practical aspect, theory aspect, any area, just come to the comment section and ask your question there. We have come to the end of the lesson. If you have gained one or two things in this video, kindly like, comment, share with a friend, and don't forget to subscribe. Meet you in the next science adventure. Remain yours sincerely, Pen with love.